Hi, everybody. I am at my house right now, and I thought I'd do a little review for you if you can't make it in for a review session, or if I don't have time for a review session. Because I'm at home, you might hear some dog barking, or you might hear my husband taking a shower, or some weird noise in the background, but just go with it. I'll do a few reviews, a few videos. I don't know how long it'll take. I'll try to make it fast because I know everybody wants to study as fast as they can. You should be able to see two screens right now. On the left, I have our immunology learning targets, and on the right, I have the three sets of notes that we've done in this unit. So I thought I'd just kind of go back and forth. Once we've done this, I feel like you should be really ready to do your study guide, and then you can quiz yourself on your study guide. This might take me more than one video because the videos are limited to 10 minutes. I can't make them any longer than 10 minutes, so I might get cut off at the end, and then we'll just go to the next video so that we can keep going. So let's go ahead and get started. I see that the first learning target says I can briefly describe the history of bacteria, including, oops, that's not working, there we go, including when they first appeared on Earth and their relation to mitochondria. So we think that bacteria first showed up on this planet about 3.5 billion years ago. So there was about 1 billion years with no life on this planet. And scientists think that mitochondria used to be bacteria. They used to be prokaryotes that moved into a larger cell. They think that because mitochondria are the same size as prokaryotes, they have their own DNA, they divide differently, they have bacterial ribosomes, all kinds of things. So they think mitochondria one time were bacteria. Oh, and do you see the number next to the learning target where it says their relation to mitochondria, and then there's a number 1? That gives you an indication of approximately how many questions I have on the test that has to do with that learning target. Any given year, I might remove a learning target or a question, or I might add them depending on what we covered during the year. So it's an approximate, it's not a for sure. Okay, the next one says I can describe the difference between the two domains of bacteria, the Archaebacteria and the Eubacteria. The Eubacteria are the ones that you come in contact with, and some of them, just like 1% of them, might cause infection, might cause disease. The rest of them don't care about you, or they don't affect you in any way. Some of them are even helpful for us. The Archaebacteria are the ancient bacteria that they tend to live in more extreme environments. And if you look at the next learning target, it asks about the different Archaebacteria. There are the methanogens that make methane. We learned about them in the digestive tracts of cows. They fart methane. There's the extreme halophiles that live in extremely salty conditions that no other life forms can survive in. And then there's the thermoacidophiles that live in the hot, acidic environments of like hot springs or thermal vents at the bottom of the ocean. And that's all we need to know about Archaebacteria, nothing more. The next learning target says I can explain that less than 1% of Eubacteria make us sick, and that Archaebacteria don't affect us at all. Because Archaebacteria live in extreme environments, and we don't come in contact with Archaebacteria. So the only type of bacteria that might get you sick would be the Eubacteria. And most of them, again, are harmless. But an infection, say Streptococcus, Staphylococcus, those are examples of, like strep throat is an example of a Eubacteria that could make you sick. All right, next, oops, I gotta scroll down here. The next learning target says I can describe the Eubacteria with the following terms. Remember, they have an outside capsule that is sticky and causes it to stick to your hands, and that's why we use slippery soap. They have a cell wall, which is unique because human cells don't have a cell wall, so it's a good source for antibiotics to attack. If they attack a cell wall, it won't affect us. They have no organelles except for ribosomes, and then they have some DNA, but they're smaller than a nucleus, so they have nothing inside them. They're tiny in comparison to eukaryotic cells. And then they have tiny little pili that can be used to exchange DNA or to help it stick to surfaces. All right, next it says I can explain how bacteria reproduce. Well, you guys, bacteria just clone themselves through the process called binary fission. They just split in half. So then the question becomes, how can they ever evolve? How are they constantly changing? And there's 3 ways that bacteria can gain new genetic information. The first way is by sucking up DNA from dead bacteria. That's called transformation, and we're on the next learning target. They can also exchange DNA through their pili, called conjugation. It's sort of like sex, only they don't have male and female. Or they can get DNA from a virus that's picked up DNA from one bacteria and infected it into another bacteria. But those are the three ways that they can evolve and get new DNA. Okay, the next learning target says I can describe how bacteria make us sick. The way that bacteria make us sick is very different than the way that viruses make us sick. Bacteria actually release toxins or poisons into our body that kill our tissues and make our organelles not work properly, whereas viruses cause our cells to explode when they get replicated inside. You might have 10,000 viruses inside of a cell and the cell just bursts open. Totally different way of making us feel crappy. All right, the next learning target says I can analyze Petri dish results and determine the most effective antibiotic by comparing zones of inhibition. So we looked at these 4 Petri dishes and we said that they've all 4 been inoculated. That's the fancy word. They've been covered in bacteria. And then they added these little discs that have antibiotics on them. The clear area around some of the discs is called a zone of inhibition. And that's an area where all the bacteria have been killed. So when you look at these when you look at these Petri dishes, you want to choose the antibiotic that has the biggest zone of inhibition. That's going to be the most effective antibiotic. And the antibiotics that are completely covered over, the bacteria is resistant to those antibiotics. So those would be the absolute worst antibiotics to prescribe for somebody. And then we looked at this one in class, and we said that we would for sure give the IPM antibiotic, that would be, it has the largest zone of inhibition. But then we also noticed that around some of these others, there's tiny little white dots. Those tiny little white dots are bacterial colonies that are starting to grow. Why are they able to grow close to these other 3 antibiotics? That's because some of them are actually resistant. And so over time, their population is going to start increasing and increasing and increasing. And if we were to do this Petri dish again, we might see that those other 3 antibiotics are no longer effective, that this particular bacteria is developing resistance to those 3 antibiotics. At this point, the IPM looks like it's like nobody is resistant to it yet. So that would be the best prescription. All right, let's see. What does the next one say? I'm trying to find it here. I can describe how antibiotic resistance happens. So that's our next section. Antibiotic resistance, if I'm going to sum it up really fast, we kill bacteria with antibiotics. So we kill the weak ones, the ones that are susceptible to the antibiotic. And then your immune system gobbles up any strong resistant ones. And that works great as long as you have a good immune system. But there are some people in our society who have weak immune systems. Old people, AIDS patients, organ transplant patients, young people, really young, like babies. They don't have good immune systems. And so if they take an antibiotic and they get a resistant form of the bacteria, their immune system might not be strong enough to eat up the remaining resistant bacteria. So then those resistant, the strong bacteria start to multiply inside of their body. And then now they have a resistant form of the infection, which means that they have to be given a different antibiotic. It also means that if one of those people with a resistant infection coughs on us, now we have the resistant form of infection and the antibiotic doesn't work on us either, even though our immune systems are normal. So we have to be super careful about using antibiotics. And we talked about that a little bit at the end, just making sure that we use our antibiotic prescriptions completely and that we finish the whole thing if we ever are given antibiotics. Okay, I believe the next section is on viruses. So I'm going to switch notes and talk about viruses. Give my computer just a second to apply. Hello, computer. There we go. All right. The first learning target on viruses says I can describe, oops, nope, that's not correct at all. I can describe the basic structure of a virus. So viruses quickly are just proteins wrapped around DNA or RNA. They are so incredibly small. So they're not even the size of a cell. So this picture here shows viruses sitting on a bacteria. So that shows you how incredibly tiny they are compared to a bacteria. Okay, I see that I'm reaching my limit for the review, so I might get cut off. So I'm just going to keep going until I get cut off. The next learning target says I can put them in order. So we'll get to that, I guess, in just a minute. We'll come back to that one. I can describe the steps of the lysogenic and lytic cycles. So the lytic cycle is how viruses make us sick. They inject their DNA. The DNA says, build more viruses.